And happy Labor Day, everyone. So, this is microbiology starting our third week. Wow. I haven't figured out exams yet, but we're working on it, and I'll let you know how things go with that. We are going to be picking up now on Chapter 4, in which we talk about some of the specific structures that bacteria have that eukaryotes don't have. I'm going to close the door here. So I hope you all have been keeping up with your reading, maybe taking some notes, taking care of things. So we were talking about some basic cell biology a bit last week and some of the structures and things that are common in both prokaryotes and eukaryotes. And then we were talking about some of the ways we can begin identifying bacteria. We're going to get into some more of these key bacterial characteristics today or this week in this chapter. So we're going back to the bacterial cell, and we have these two defining regions that are critical in bacteria to talk about. First, we've got, we're going to separate them by the plasma membrane, which of course is the phospholipid bilayer, and the phospholipid bilayer is made up of phospholipids. These are these amphipathic molecules that have a phosphate head and two fatty acid tails. And so the phospholipid bilayer is arranged like this. And it's a bilayer, meaning that we have two sides of it. So the external environment that organisms find themselves in is largely aqueous, watery, and so the charged head is facing that side. And then the cytoplasm is also largely watery and polar, and so the charged head is facing that side. So that's the plasma membrane. And then in bacteria, we have these two different kinds of regions that we refer to. One is the region known as the envelope. And this is everything from the plasma membrane out. As we're going to start seeing here, there are a lot of candy coatings and walls and things on the outside surface. So this is the external environment. And we've got the envelope. So the envelope is everything from the plasma membrane out. And then we've got, on this side, the cytoplasm. And the cytoplasm is the term we use for everything on the inside of the plasma membrane. And this includes the region known as the nucleoid, which, while it's not a membrane-bound nucleus like in eukaryotes, it is the spot where the DNA is arranged and where transcription is also taking place, and it's located in the cytoplasm. So a couple of things about the cell membrane. The cell membrane serves as a barrier. The cell membrane is essentially a boundary layer that is able to exclude things from moving in or exclude things from moving out. It's a way you can begin to accumulate stuff. So, just to keep up with this drawing here, our central spot for our organization of the envelope versus the cytoplasm is the plasma membrane. And the plasma membrane is also the site of a variety of different kinds of proteins. And we can have proteins that are what we refer to as being transmembrane proteins. And then we can have proteins that are peripheral, or on the surface, peripheral proteins. Now, the way you determine experimentally whether a protein is peripheral or transmembrane is peripheral proteins can sometimes be knocked off by washing the cells in a high salt solution, whereas a transmembrane protein, the only way you can liberate those proteins is to destroy the plasma membrane, and typically by adding detergents. And you can solubilize the plasma membrane away, and you can have then in soluble form these transmembrane proteins, because the middle of the transmembrane protein is typically going to be hydrophobic or water-fearing. It's going to have a set of amino acids that are nonpolar. Now, the plasma membrane does a number of key things. It is serving as essentially a boundary layer, and there's an A in there, a boundary layer, and that means that we can allow, sort of as a gatekeeper, material to move in or material to move out. And so things can go in and out of the cell by a variety of different mechanisms. We can have things like water and oxygen and CO2 move back and forth through what's known as passive diffusion. Some nonpolar molecules also can move through this way. Other things that are going to have a bit of a charge to them can be moved in using a transport protein in something that's known as facilitated diffusion. Now, this is a term that I guess I kind of expect you to have picked up somewhere. Diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. So it is just a physical phenomenon, like when you add a drop of dye to water and you watch it move out, or you open up a bottle of perfume in a room and it begins to diffuse around. That's passive diffusion. Now, in cells, if you've got a molecule that's got a charge to it, it can't diffuse through the plasma membrane by itself. Small nonpolar or reasonably nonpolar molecules, or molecules that don't have much of a charge, like water and gases, can diffuse through the phospholipid bilayer, sort of wiggling their way through. But molecules like sugars, amino acids, all the different kinds of charged ions and things, they can't move through the plasma membrane, but they can move with their diffusion gradient, meaning they can go from an area of higher concentration to an area of lower concentration, in or out. And they do that through specific protein channels that are known as passive transport channels. So this is facilitated diffusion. Now, another kind of facilitated movement is something that is known as active transport. So, active transport is now actually going against the diffusion gradient. It's accumulating things one side over the other. So, say you want to begin pulling in lots of sugars and generate a concentration of sugars higher inside than outside, diffusion won't work. So, there are proteins, these active transporters, that actually can bind to glucose and change their shape, put the glucose in, and the glucose typically, or the sugar, gets modified in some way that won't get diffused back out. It won't recognize the transporter again. And so, these are ways we can begin moving molecules around the plasma membrane. We've got these different kinds of barriers here, and we have all sorts of different kinds of transporters, and pretty much any kind of charged molecule and any sugar is going to have a transporter. Sometimes it can be just a channel that allows things to come through, other times it's an active transport system. So the cells are quite busy all the time maintaining their salt and sugar concentrations. In animal cells, we spend, you and I, up to 30% of our calories that we consume in a day are doing nothing but maintaining sodium and potassium concentrations. We always depend on having a high sodium concentration outside of the cell and a low potassium concentration outside of the cell. So we're pumping sodium out and pumping potassium in. And this actually requires the expenditure of energy in the form of ATP. And you spend just sitting around the couch. What are you doing? Well, at least you know you're pumping sodium out and potassium in, and you actually use a fair amount of calories doing just that. That's how critical these kinds of transport systems are. So bacteria are doing that as well. Now, when we start thinking about bacteria specifically, they've got a very complicated cell wall interaction. Now, you and I don't have cell walls. You think about your red blood cells. One of the tests for red blood cells is if you put them into water, they actually will swell up because of diffusion. They got a lot of salt inside, water moves in, and very quickly they'll pop in the lyse. Now, most of our cells are sensitive to that, which is known as osmotic stress or osmotic movement. And we've got a variety of different kinds of structures and things that try to keep the fluids from moving in and out too fast. We maintain a certain salt concentration within our bloodstream. We've got a fairly tough coating around us, but there are a variety of cells that actually are going to be rather sensitive to water concentrations and water movements. Plants, on the other hand, of course, have the cellulose-based cell wall. I think of plants sort of like having a balloon in a shoebox. And they can pressurize themselves up. As they pull water in, they can pressurize against the shoebox, the cellulose wall. And that's how plants stand up after you give them a little drink of water. So plants are always under a positive pressure, and they're pushing out against the cell wall. Bacteria are doing a similar kind of thing. They don't have a cellulose cell wall, and they're certainly not organized into a large cell structure like a plant is. They're being single cells, but they do have a cell wall type material which keeps them from popping when they're in a really wet environment, which is often how they're growing. So the the bacterial cell wall comes in two different types, and we can categorize bacteria on their cell wall type. And this is a term that perhaps you've heard, and when we start thinking a little bit more about doing our virtual lab, we'll be looking at some of this stuff. But the terms that we've got for for bacterial cell walls are denoted by a specific kind of stain that is called a Gram stain, named after Mr. Gram back in probably the 1800s. And we've got these two terms. One is called being Gram positive, also indicated as being Gram plus. And then we've got a cell wall type that is, happily, Gram negative. And these are two very distinct different types of wall materials and wall organizations. And they kind of are doing the same thing, is they're keeping the plasma membrane from rupturing as water moves in, and it maintains a certain pressure inside the bacterial cell wall. Let's look at a couple of pictures here, and I think we can describe it a little bit better than going on the board. There's some structures and things, and there's some very interesting things about the bacterial cell wall that we want to get into. So hopefully, using the PowerPoints, if this works for you as well, taking notes off the PowerPoints is a useful thing to do because, as I say, writing stuff down still really does work to actually put it into your head a little bit better. So we've got now a diagram, and you can find something similar like this in your book. In that, let me get my pointer going. Beep. we've got the gram positives and the gram negatives. So gram negatives are a bit more complicated than gram positives. And part of what these guys are also doing, these walls, is they're providing compartments. We've got the cytoplasm down here, and then we've got now a periplasmic space, a compartment that is not in the cytoplasm, but it's not completely external. And this is allowing us to actually do some interesting things when we start coming to pumping ions around. And the gram positives are coated with a single molecule, more or less, of something called lipoteichoic acid. It's exactly not what it is a fatty sugar polymer. Not critical that you memorize any structures or anything, but it is known as the peptidoglycan layer. I would write that on the board, but I ain't got no board space. So the peptidoglycan layer is made up of sugars and proteins inside it. So this is part of what makes the bacterial cell wall somewhat slippery and actually have a very specific characteristics of a smooth candy coating. Now, if we look to a gram negative, we see gram negatives are a little bit more complicated. They've got an inner membrane, the plasma membrane, and then here are the different proteins and transmembrane proteins and such. They also have a periplasmic space, like right there. But now instead of having just a simple peptidoglycan layer, there's peptidoglycan in the periplasmic space, and then we have this outer membrane. And the outer membrane contains a variety of different kinds of molecules. Here is a collection of proteins known as porins, which are pretty big tunnels through the through the outer membrane layer. We've got a whole range of not necessarily phospholipids, but what are known as lipopolysaccharides. Now, lipopolysaccharides are, kind of as the name implies, they have a fatty acid tail, and then instead of having the phosphate and the decorative choline molecule, they've got a whole stretch of sugars, of long-chain sugars, lipopolysaccharides. Now, these actually are doing similar kinds of things as the gram-positive peptidoglycan layer in that they're sticky and that they're able to interact with the surfaces, but they are a bit more complicated. Now, both gram-positives and gram-negatives have this thing called peptidoglycan. And if you're into comparative structures and things, animals have kind of their own sugar coating that cells are embedded in, particularly like in different tissues. And these are things that are known as glycosaminoglycans. And it's a similar kind of molecule, and the glycan is a sugar. And they have different sorts of decorative groups. So we've got a couple of different kinds of sugars here. We have N-acetylglucosamines, NAGs, and we have those as well. And then N-acetylmuramic acid, NAM. These are just names for different sugar polymers. And they have amino acid protein bits coming off of them, and then they also have cross-bridging, and they can form this very tight mesh, kind of like a chain link fence almost, of sugar with these small proteins coming across it. So this provides this really tough net. And because the structure is unique to bacteria, it is a really good target for a variety of antibiotics. So there are a number of antibiotics that target the production of the cell wall, of the peptidoglycan cell wall. Things that you know of for sure, like the penicillins, which is ampicillin, amoxicillin, cephalosporin. Let's see, a couple other esoteric ones, cycloserine, vancomycin, not used very often. They're also involved in blocking the cell wall formation. Bacitracin is another actually fairly common bacterial antibiotic used for treating sinuses and things. And what they're doing is blocking the bacteria when it divides from producing new peptidoglycan. And if it can't produce new peptidoglycan, the cell is going to be subject to osmosis, and it's going to swell up and pop. Now, one of the points behind this is if you've got a really bad bacterial infection already, giving antibiotics is going to actually cause a lot of bacterial lysis and a lot of bacterial debris and toxins to be released. And so immediately after antibiotic treatment, if it's a really bad bacterial infection, can be a bit of a dicey time, but hopefully you make it through that period and then you can actually inhibit any further bacteria. So this is a structure, not that we need to memorize this in any way, but this is penicillin. And it's the general shape of this. This is known as the lactam ring right here. And bacteria that are able to grow in the presence of penicillin or ampicillin produce an enzyme called beta-lactamase. And they break down this lactam ring and they render the penicillin or the lactam ring based antibiotic non-functional. And beta-lactamase is an enzyme, it's a protein. And so bacteria can transfer the gene for beta-lactamase among friends, and now if you've got that gene, you can be resistant to antibiotics or to this specific antibiotic. So this is one of the ways that we can think about bacterial antibiotics is we want to target things that are not specific to eukaryotes. We want to be able to kill the bacteria, but not the host. So a bit more on lipopolysaccharides. They are really important molecules to think about because they actually have a lot of they're responsible for a lot of bacterial-based complications. So they're on the outer surface of gram-negative cells. So lipopolysaccharide is not present in gram-positives. Now, the really critical thing about lipopolysaccharide, also abbreviated as LPS, and you may see this in the literature sometimes, is it is very antigenic. The immune system responds to it like crazy, and it is able to generate a really big immune system response. Lipopolysaccharide is also responsible for something known as endotoxin. Endotoxins are a specific class of lipopolysaccharides that can be released from certain bacteria. And endotoxins are going to overreact or cause an overreaction by the host's immune system. And so you can create tremendous inflammatory response and all kinds of things like toxic shock syndrome, a whole range of physiological phenomena that occur because of the large bacterial infection. And so there are some bacteria that have very potent lipopolysaccharide endotoxins, and even having a few of these bacteria can be really damaging. So here is the overall structure of a lipopolysaccharide. We've got the fatty acids. They're linked in a slightly different way than they are in a phospholipid. And we've got a disaccharide phosphate. So there's a phosphate here and here. This is going to be a very charged head, and then this is going to be the non-charged, non-polar tail. And then we've got a whole range of different kinds of sugars that are repeated in a polymer-like way sticking out the top. So it's a fairly large molecule. It's got a lot of sugars on it, and it's able to interact with the immune system in ways that are not particularly friendly. So it is able to generate a large immune response. Lipopolysaccharide, LPS, is something that can be used to generate a large-scale sort of immune reaction. You know, not specific, but it just cranks up your immune system. And that can actually generate some pretty serious problems. So now, another term we've got, and This is a little bit easier just to go with PowerPoint than write it on the board. So excuse me if I do this, but you'll survive. What you should be doing, though, is writing out your vocabulary list. We have and this is sort of the same term for some of the components of this candy coating. It can also be known as glycocalyx. And the glycocalyx is the term we use for this gel coat around the bacteria. Now, if it's very closely attached, it's referred to as a capsule, and you can see this under a microscope. It's actually pretty tightly bound, and you don't really see anything outside of the bacteria itself. If it's not tightly attached, it actually can kind of ooze out, and it's referred to as a slime layer. Ugh. Now, we've mentioned this before. If you've got like a slippery rock coated with bacteria, very often it's got the slipperiness coming from this slime layer, which is all this sugar and material being dumped out. Now, one of the things this does is it is providing a sticky surface, almost like tape, that keeps the bacteria attached to surfaces. That's why washing your hands is a critical thing to do to remove bacteria. They can't be just wiped off very easily. It helps to keep the cell hydrated because there's a lot of hydrogen bonding capacity here that it can hold lots of water molecules. And it also seems to protect the bacteria against the immune system. The immune system is not necessarily able to recognize this very well, bacteria that have these capsule layers, and it's sort of a stealth protection. So we got the lipid bilayer. We've got some glycolipid. This is fatty acid tail hooked to a sugar chain. We've got a variety of proteins that are known as glycoproteins. It's one of the things we didn't really hit on when we were talking about the different biopolymers, but we can begin to mix and match, And so you can have, very often, we have proteins that have sugars coming off of them, glycoproteins. Most of the proteins in our body, certainly all the proteins that are on the cell surface of a cell, are all glycosylated. They have sugars attached to them. And these do all kinds of different kinds of functions and also provide bonding interactions between cells. So we've got a transmembrane glycoprotein. We have a peripheral glycoprotein. Then there are some of these molecules that are mostly sugar, but with a small protein core, and those are known as proteoglycans. We have glycoproteins, which is mostly protein and some sugar, and then we've got proteoglycan, which is mostly sugar and some protein. A proteoglycan that you're quite familiar with is the active ingredient in Jell-O. Jell-O is made up of proteoglycan. And what that means is that when you heat it up, the sugar molecules are able to kind of open up, and then as they cool, they hydrogen bond with each other, these sugar polymers, and you get the gelling effect. So these are all part of this capsule slime layer material. Now, another part of the bacteria system that is important to think about and useful in terms of structures that are different from eukaryotes is different kinds of appendages. Now, animal cells and plant cells can contain cilia, and certainly in flagella, lots of microorganisms, protists that are not prokaryotes, they can contain cilia and flagella. But bacteria have a very specific kind of structure that is specific to bacteria. And so the flagella are built with a whole range of proteins, and you don't need to in any way learn the different kinds of structures here. It is a series of proteins that are able to hydrolyze ATP and begin spinning. And when they do, we've got a filament that is actually asymmetrically mounted, and it begins to whip, kind of like a screw or a propeller, and it can begin to actually move the bacteria around. Now, another kind of appendage is something known as pili or pili. These are long strands of protein, and they allow for attachment to a variety of different surfaces. So you got this stringiness coming out of this glycocalyx peptidoglycan layer, and that allows for the bacteria to kind of entangle themselves. We also have the sex pilus for conjugation. This is more or less bacterial sex, kind of. It is a way of two bacteria to transmit DNA from one to another. We have pili on the bacterium that causes gonorrhea, Neisseria gonorrhea, that allows it to anchor to epithelial cells. And so these external things are either serving as a stealth mode to keep the immune system from binding to them, or they're serving as attachment points, so they can do a variety of different kinds of things. So now, the last bit for the day is into the cytoplasm. And we've got a couple of formed elements in the cytoplasm of prokaryotes. Now, one of the points, again, as we think about eukaryotes, is that eukaryotes have got a lot of different subcellular structures. We've got the nucleus, we've got the nucleolus, we have the Golgi, we've got all kinds of different vesicles. Bacteria tend to be a bit simpler. Now, one of the things we can see, and pretty much you've got to use electron microscopy in order to see these things, but we've got ribosomes. Ribosomes we'll be talking about as we get into some of the protein translation machinery. Ribosomes are responsible for taking RNA and translating it into protein. Another thing that bacteria often have is a general term, something called an inclusion body, which is a granule of sorts that can store a variety of different kinds of molecules, generally different proteins. And so you can have inclusion bodies of proteins that can be accumulating in there. This term called a mesosome that's shown on the slide here is actually an artifact of electron microscopy. As we said, electron microscopy requires that the samples be treated with some rather harsh materials and dehydrated completely, and you can generate some strange structures that are in fact artifacts. So mesosome is one of those. It's not really something that's there. Okay, so the cytoplasm is mostly this kind of a gooey layer, a gel layer, that is where all the different kinds of biochemical reactions are occurring. We're going to stop at this slide and continue on from here on Wednesday. I'm going to post the PowerPoints here and start thinking about how we can give you sort of some quiz questions to think about. Mostly we're getting into this is basic cell biology stuff, a little bit of bacteria specific discussion here. And we'll see how things go from there. All right, so from a quiet Monday morning, or actually Monday afternoon, this is Dr. G signing off. In better contrast, I'll see you cats later.